Today now first topic in that course, that is the pest of paddy. So paddy is the major agronomical field crops. 1/3 population, more than 2/3 population of India having a most important and staple food, that is the rice or rice sativa or paddy, and more than 65% world population depends on the paddy. So this paddy crop is mainly infected by various insect pests. Near about more than 100 insects recorded on that crop. So out of that, 20 insect pests are major economic significance, and these pests mainly classified as a borer pest, sucking pest, defoliator pest, non-insect pest. So borer pest, in that mainly yellow stem borer, gallmich, or also called as a gallfly. Then sucking pest, in that green leaf hoppers, white leaf hoppers, brown plant hoppers, mealybugs, earhead bugs. These are the major sucking pest of paddy. Then defoliator pest means mainly that pest feeding on a foliage or on the leaves. So in that leaf roller, horn caterpillar, paddy swarming caterpillar, yellow hairy caterpillar, caseworm, paddy skipper, climbing caterpillar, Ipterotid hairy caterpillar. These are the major defoliator pest. Also grasshoppers, in that two species, Herioglypus banyan and Oxa nitidola, furl maggot, blue beetle, hispa. These are the defoliator pest. The non-insect pest mainly paddy mite, panicle, rice mite, rice field crab, and field rat. So these are the major non-insect pest of a paddy. Mite, two species of mites, field crab and field rat. The next, that is the contribution of that various insect pest as well as non-insect pest in the losses of a rice. So in that major contribution, that is the 29% of a stem borer, then 16% yield loss due to the brown plant hopper, then 14% yield loss in a paddy due to the non-insect pest or other pest, then 13% due to the gallmich or gallfly, then 10% yield loss in the paddy due to the leaf folder, then 9% due to the white backed plant hopper, 9% due to the green leaf hopper. So these are the major economic losses due to the various stem borer, foliage feeder, or defoliator or sucking pest. In that, stem borer having a first or major pest, then brown plant hopper, gallmich, leaf folder, white backed plant hopper, and green leaf hopper. So in that first yellow stem borer, Sarcophaga incertulans, family Pyrostidae, order Lepidoptera. So this pest is mainly distributed in Afghanistan, Bangladesh, Burma, that is the Myanmar, Cambodia, China, India, Sri Lanka, Indonesia. Host range, that is the only one host, that is the paddy. So why it is called as a yellow stem borer? Because adult of that pest, which is a yellowish brown color, that's why it is called as a yellow stem borer. Stem borer that is the yellow stem borer, that is the common name of that pest, because this pest mainly infests to the stem of a paddy. That's why it is called as a stem borer. And yellow because their color of a adult moth, which is a bright yellowish in color, and their forewings bright yellowish brown in color. And in that male and female distinguish in that female having a two black spots on their forewings, and in male yellow stem borer without that black spots on their forewings. So this is the major identification of that yellow stem borer pest, male and female. Then damage symptoms, mainly the larva is the damaging stage of that pest. Why larva is the damaging stage? Because this dead heart symptoms and white ear head किंवा dead panicle symptoms mainly seen in the paddy field due to the infestation of a. आवाज येतोय का? हॅलो.
येस सर, आवाज येतोय, स्क्रीन दिसत नाहीये.
हो का? इंटरनेट हे झालं का? दिसते का?
Yes sir.
In that yellow stem borer, mainly two types of symptoms found due to the infestation of a larva. First, that is the dead heart symptoms, and second one, that is the white ear symptoms due to the infestation of a larva. So, dead heart symptoms mainly found due to the infestation of that larva, particularly on the stem. So initially that larva bore the stem with the inner content of that infested stem, and due to that drying of the central shoots, that is called as a dead heart in the young seedling stage. Then second one symptoms, that is the drying of the panicle in a growing plant called as a white ear. White ears in that so this type of white ear symptoms due to the infestation of yellow stem borer larva. So in that drying of the panicle in the growing plants, and dead heart symptoms, that is the drying of the central shoots, and central shoots becomes a dead, and that symptoms by in the white ear symptoms forms in the growing plant, and dead heart symptoms mainly noticed during the seedling stage. So grown up plant totally whole panicles becomes dried, that is called as a white ear. Then identification of that pest. So mainly their eggs. Each female deposit near about 170 to 200 eggs in a groups of 50 to 80 on the upper surface of lip tips covered with buff colored hairs. So these are the flat in shape, oval in shape, and scale like and laid in a groups, and these eggs mostly covered with a buff colored hairs. So this is the this yellow stem borer mainly deposit their eggs specific on the lip tips of a upper surface of a lip tips. So that is the specific site of oviposition of that yellow stem borer, upper surface of lip tips. So this is the specific site of oviposition of yellow stem borer. Each female deposit how much eggs? 170 to 200 eggs in a groups, and then these eggs should be covered with hairs for the protection of natural calamities, biotic factors. So specific site of oviposition, that is the lip tips, upper surface of a lip tips. There they should oviposit their eggs. Then larval stage, mainly larva which is a pale yellow in color, and head of that larva which is a dark brown or blackish in color. So pale yellowish type ची त्याची larval stage असते. Larval period is near about 20 to 45 days. Then third one stage, that is the pupal stage of that pest, which is a pupa of that pest, which is a white silken cocoons found inside the stem. Stem chart मध्ये white silken cocoons मध्ये ह्यांचा हा pupal stage असते inside the stem, and pupal duration of that pest is near about 6 to 7 days. Then fourth one stage, that is the adult. In that female moth and male moth. So female mainly bright yellowish bright yellowish brown in color, bright yellowish brown forewings with a black spots on their forewings. And in the male comparatively smaller in size as compared to the female, and males forewings without the black spots. So we can easily distinguish that male and female adults of yellow stem borer on the presence of the black spots on their forewings. In female, there are the two black spots on their forewings, and in male these forewings are without a black spot. So this one is the larval stage, mainly pale yellowish in color and dark brown head. Then ETL level means economic threshold level of that pest, that is the 2 egg masses when observed per meter square area. At that time we should initiate the control measures, or 10% dead herd symptoms should be found at the vegetative stage. Then 2% 5 years in the flowering stage. So at that time we should initiate the application of various management practices. So according to that ETL level, we should apply the various management practices. 2 egg masses per meter square, or 10% dead herd symptoms in vegetative stage, or 2% 5-year symptoms in a flowering stage. So in a bionomics or life cycle, in that generally female moths deposit their eggs mainly on the upper surface of a lip tips. they deposit near about 170 to 200 eggs. Then larva will be comes out within a 5 to 6 days from that eggs, having a 5 stages of that larva. Larval period is 20 to 45 days depending upon the temperature. Then pupa of that pest mainly found inside the infested stems in a white silicon cocoons. Pupa which is a dark brown in color. Pupal period is 6 to 10 days. Then adult stage of that pest which is comes out from the pupa, and total life cycle of that pest is completed within a 28 to 35 days depending upon the temperature. So this one is the female adult moth of yellow stem borer, having two black spots on their forewings and forewings which are bright yellowish brown in color. Then in that yellow stem borer, male and female adults. So male comparatively small in size and without black spots on their forewings. Female which is comparatively bigger in size, forewings are bright yellowish in color and prominent black spots in the center of each of the anal end of the forewings, and anal end with a tuft of yellowish hairs of a forewings. Here is the anal end of that forewings where the yellowish comparatively yellowish hairs which are much more as compared to the male yellow stem borer. Then these are the damaging symptoms. So this is the yellow stem borer larva mainly infest to the stem portion, feed the inner content, and due to that infestation, dead hearts or drying of a central shoot during the vegetative stage. Mainly this yellow stem borer infestation noticed on a deep water rice. So it is found in aquatic environments where there is a continuous availability of a water or continuous flooding of a water. Second instar larva mainly enclose their body in a leaf wrapping to make tubes and detach themselves from leaf and falls onto the water surface, and thence they enters into a stem. Mostly high nitrogenous field favors population built up of a stem borer. जास्त प्रमाणात जर आपण नायट्रोजनस फर्टिलायझर्स अप्लाय केले तर तिथं त्या येलो स्टेम बोररचे इन्फेस्टेशन हे सिव्हिअर असते. Fields planted later favors more damage by the insect pests that have built up in a fields have been planted earlier. लेट प्लांटिंग जर झालं तर त्याच्यावरती जास्त प्रमाणात त्याचा इन्फेस्टेशन असते as compared to the early planting. How to identify the infestation of that pest in a field conditions? So check the field regularly. Dead hearts or dead tillers should be pulled out and see the infested portion of that stem having any larval stage of that pest. White heads or white ears symptoms also seen in a reproductive stage or panicle formation stage. Tiny holes also seen on the stem as well as tillers. Frost or fecal matters means their excrete of that larva inside the damaged stem. So these are the signs of the infestation of yellow stem borer. Dead heart, white ear, fecal matter, these are the major signs of the infestation of yellow stem borer. Then why it is important, why this pest important? Because excessive loss, minimum 20% yield loss in a early planted rice and 80% in a late planted paddy crops. So that's why this is the very serious pest of a paddy. Maximum 80% loss noticed due to the yellow stem borer. So these are the drying, central shoot, drying, dead earth symptoms, and white ear panicle symptoms, mainly due to the infestation of yellow stem borer. So these are the white ear symptoms in a paddy during the reproductive stage of a paddy. These white ears are totally empty ears, or their grain fill up percentage as comparatively healthy panicles is very less. How to manage? In that, management practices: first one, application or grow resistant varieties. In that, Ratna, Jaya, TKM6, IR20, IR26, Sai Sri, Saket, IET 3127, IET 2812, MTU 5849, PTB12, PTB20, PT321, H4. These are the resistant varieties against yellow stem borer. Then this is the mechanical practice. In that, clip the seedling, clip the seedling tips before transplanting. Before transplanting, we should clipping the seedling tips, because they mainly prefer that upper surface of leaf tips for the deposition of their eggs. That's why clipping of leaf tips before transplanting. Then avoid close planting or and continuous water stagnation at early stage. So these are the favorable conditions for the multiplication of that pest. Then collect and destroy dead herds, white ears from the field along with the larva. Set up light traps to attract and kill the moths. Then install 6 pheromone traps to monitor and mass trapping of a adults of a yellow stem borer. Then this is the specific eggs parasitoid for the yellow stem borer of rice, that is the Trichogramma japonicum. They mainly parasitoid the eggs of yellow stem borer. So that's why it is called as a egg parasitoid. Should be released 2 times on 30 and 37 days after transplanting, at the rate 5 cc per hectare per release. So this is the release of a Trichogramma japonicum eggs. Eggs card of that Trichogramma should be stapled in a field, and from that adult of that Trichogramma comes out and they parasitize the eggs of a yellow stem borer. So 5 cubic centimeter eggs per hectare per release should be 5 cubic centimeter, 5 lakh cubic centimeter eggs should be released per hectare. So this is the Trichogramma japonicum, which is the specific eggs parasitoid of a yellow stem borer. Then application of Bacillus thuringiensis, that is the BT variety. In that, crustiki and neem seed kernel extracts in the combination of 2.5 gram per liter and 1% to reduce the oviposition of a stem borer. So this is the Bacillus thuringiensis plus neem seed kernel extract, which is helpful for the prevention of a eggs deposition by the yellow stem borer. Then application of various chemicals in that, mainly granule formulation chemicals, which is directly we should apply in a particular infested paddy plant in their whorl. So carbofuron 3G at the rate 25 kg per hectare, then benfuracarb 33 kg, chlorotranilprole 0.4% granule 10 kg, fipronil 0.3% granule 70 to 25 kg, cartap hydrochloride. These are the granule formulation chemicals effective against yellow stem borer. Then application of various chemicals in a spray formulation: acephate, cartap hydrochloride, monoprotophos, quinolphos, azadirachtin, carbosulfan, chlorantraniliprole, ethofenoprox, fipronil, flubendiamide. So these are the liquid formulations of that chemicals also effective against yellow stem borer. Flubendiamide, lambda-cyclothrin, phosphomidone, thiacloprid, thiamethoxam. These are the effective chemicals. Then harvest the crop up to the ground level and disturb the stubbles with the flowing immediately after the harvest, because their larva should be kept. The larva should be remains in that stems or stubbles uh, which is remaining after the harvesting of a paddy field. So harvest the crop up to the ground level and disturb the stubbles with flowing immediately after the harvest to killing the larval as well as pupal stage of that pest. The next, that is the major pest, that is the gall midge. So midge or fly, this is the Dipteran order pest family Cissidomidae. So mainly host range rice, wild rice of Oryza grasses, various species of grasses. So these are the eggs, then maggot and adult stage of that gall fly. So mainly their adult, which is looks like a mosquito, which is orange in color. Then various life stages of that gall midge or gall fly. In that first one, eggs. So mainly eggs which are tubular or elongated in a shape, whitish in colors. So near about 100 to 300 eggs, mainly deposited by the adult of fly near the ligule of the leaf blade. So that is the specific site of oviposition of that gall midge. Mainly they deposit their eggs on the just near the leaf, just near the ligule of the leaf blade. So after the hatching from that eggs, that maggot initially start their infestation on the stem. That's why they deposit their eggs near the ligule of the leaf blade. Then second one, a stage of that pest, it is the maggot, which is near about 1 mm long in size. It creeps down the sheath portion of a plant and enters the growing buds, and oval chamber is formed around the site of feeding. Maggot mainly pale red during the feeding, and larval period is near about 8 to 10 days. Then next one stage of that gall midge, that is the pupal stage. Maggots pupates at the base of the gall and moves to the tip of the gall and project outside during emergence. So pupal stage of that gall midge at the base of the gall form at the site of their infestation. Then adult mainly yellowish brown looks like a mosquito. The male, which is ash gray in color, adults feeds only on a dew drops. So their infesting stage of that pest is the only maggots. So this one is the adult stage. These are the eggs. That is the this one is the pupal stage of that pest. Life cycle in that there are the 4 life stages: eggs, larva, pupa, and adult. So mainly adult deposit near about 100 to 300 reddish elongated tubular eggs just near the ligule of a leaf blades. Then eggs period is near about 3 to 4 days adult. After that, maggots will be comes out from that eggs. Maggot period is near about 8 to 10 days. Then maggot goes to the pupation at the base of a galls. Pupal period is near about 6 to 7 days, and total life cycle of that pest is completed within a 15 to 20 days. So mainly their site of oviposition, that is the ligule of the leaf blade, where they deposit 100 to 300 eggs, and their pupal stage or pupa of that pest at the base of a gall. Then nature of damage. This is the diphtheria order pest. Mainly their damaging stage is the maggot. So mainly maggots after hatching from the eggs feeds at the base of the growing shoot and cause a tube-like formation at the base of a shoots, that is called as a silver shoot or onion shoots. So mainly that maggots which is feed on the growing portion of the plant, growing portion of the shoots, and due to that infestation they forms a tube-like structure or gall-like structure. That's why it is called as a gall midge or gall fly. So due to the infestation of maggot, their cells becomes elongated and forms a gall-like structure, and due to that infestation the growth of the tiller should be arrested or stopped. So the feeding by maggots and their larval secretion, which contains an active substance called as a cystidogen, which is responsible for the cell proliferation of the meristematic cells. As a result, gall formation takes place. So mainly due to the infestation of that gall midge, that is the gall formation. Gall formation means excessive growth of the leaf tissues. So this gall formation takes place due to the chemical substance which is secreted by that maggot, which is called as a cystidogen. So due to that infestation, the growth of the plant is totally arrested. The galls are formed by the gall fly are nothing but that is the modification of a leaf sheath. So actually that is the gall formation, that is the modification of a leaf sheath due to the infestation of a maggot. They secrete a chemical substance, that is the cystidogen. The central shoot, instead of producing leaf, produce a long tubular structures called as a gall. So due to the infestation of that maggots, that infested shoots does not produce a leaf or leaves, and instead of that they produce a tubular structures called as a galls. And when gall elongates as an external symptoms of a damage. So infested tillers are not producing the panicles. So generally, this type of symptoms forms due to the infestation of a gall midge. Silver color of a shoots or onion shoots are also called as a silver shoots. That is the main external symptoms we can identify if the infestation is done by the gall midge. Tubular galls, tube-like structures of that infested paddy field in a tillers. So damaged tillers turns into a tube-like galls, which latter becomes dry without a panicle. Then these are the main external symptoms, that is the silver shoots or onion shoots. And due to that, due to such type of damage, the panicles does not, the tillers does not produce the panicles. So there are adverse effect on the yield. So at in that infested shoot, we can see the maggot of that gall midge, which is mainly yellowish in color inside the base of the stem portion of that paddy. Then regions, which are the major regions in that early monsoon showers. uh, Main reason of that infestation: early monsoon showers following by a longer dry spell, continuous cloudy weather associated with intermittent rains, means without continuous rains their infestation will be increased. High relative humidity, optimum temperature range 28 to 30 degrees centigrade, late planting, high yielding susceptible varieties, continuous cropping, high nitrogenous application of fertilizers, close planting, intensive application of management practices. These are the major reasons for the outbreak of a gall midge infestation. Management practices in that early planting is helpful for minimize the infestation of that pest. Use resistant varieties MDU3, Shakti, Vikram, Sureka, IR36, Dhanlakshmi. These are the important resistant varieties. Flowing immediately after the harvesting of a crop, remove the alternate host, application of fertilizers in a balanced manner, set up light traps, one light trap per hectare for monitoring purpose. So these are the infrared light traps mainly useful for the monitoring of the infestation of gall midge. Then use the larval parasitoid Platygaster oryzae. Throws parasitized galls at 1 per 10 meter squares in the male field after the 10 days transplanting. Platygaster oryzae, this is the larval parasitoid. Conserve spiders like Tetragnatha, Argiope, Catenolata, carabid beetles in a rice ecosystem. Application of various chemicals in a granule formulation, in that carbofuran 25 kg, fipronil 16 7 to 25 kg or spray application of phenolphos 1 liter, ethophen fox 500 to 750 ml, fipronil 1.5 kg, lambda cyclothrin 500 ml, thiamethoxam 100 gram in partial liter in 500 liter of water per hectare. So these are the effective chemicals for the management of gall midge. So gall midge is one of the important major pest of a paddy. The damaging stage, that is the maggot, mainly cause two types of symptoms: silver shoot, also called as a onion shoots, and formation of a cystidogen in a formation of release the cystidogen chemical substance at the time of infestation, and due to that gall formation is takes place, and there is no panicle form in the infested tillers. Okay, now today's lecture is over. If anyone having a doubt, please ask. ठीक है.